Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Richard Parker-Manuel from OXGENE, a WuXi Advanced Therapies company.
Hello and thank you for joining me. My name is Richard Parker-Manuel and I am the Group Leader for Plasmid Engineering and Production at OXGENE, a WuXi Advanced Therapies company. In March 2021, OXGENE was acquired by WuXi AppTec to become part of WuXi Advanced Therapies. Together, we provide end-to-end support to cell and gene therapy companies to help them accelerate time to market. In the first part of my talk, I will tell you about our adeno-associated virus and lentivirus production plasmids, and in the second part, I will tell you about the high-quality plasmid manufacturing platform at WuXi ATU. Our recombinant AAV production system consists of three plasmids: a modified RepCap plasmid, about which I will tell you more in a moment. Then we have the transfer plasmid with the therapeutic gene expression cassette, flanked by the AAV ITRs, and finally the helper plasmid, which encodes the adenovirus genes needed for AAV production. Our helper plasmid is simplified and contains only the essential genes, whilst removing other non-essential but toxic genes such as hexon. This not only increases helper plasmid yield, but is also an advantage when it comes to regulatory approval. Now I'm going to tell you about our RepCap plasmid in a bit more detail. Rep and Cap are two open reading frames that are translated to produce multiple distinct proteins needed for AAV assembly. When we began optimizing our system, this was the first thing we looked at. As you can see, we tried various different configurations and found that CMV Cap EMCV Rep gave a significantly increased yield. Cap is placed under the control of a strong CMV promoter, while Rep is expressed at a lower level via EMCV internal ribosome entry site. Next, we used the new RepCap to produce various serotypes in a stirred tank bioreactor and achieved titers of up to 3.7 times 10 to the 11 genomes per mil. The new system improved not only production but also quality, with higher infectivity and a greater percentage of full capsids, as shown on the right. The new RepCap plasmid is also safer. Studies have shown that using a heterologous promoter such as CMV reduces the chance of generating replication-competent AAV particles. Here is an illustration of our transient lentivirus system. It is a third-generation system with the various functions split into four different plasmids. First, we have the envelope plasmid encoding the VSV-G gene, which has been codon optimized and is driven by a specific promoter intron with improved expression characteristics. Second, we have the transfer plasmid encoding the therapeutic gene cassette, flanked by long terminal repeats. As with other third-generation systems, it is replication incompetent and self-inactivating, making it relatively safe. Third, we have GAG-PUL, which has intentionally not been codon optimized as it encodes several different polypeptides whose expression may be negatively impacted by codon replacement. The fourth plasmid encodes the regulatory protein REV, which regulates transcript splicing and nuclear export. We use the combination of hybrid promoters and introns to fine-tune the relative expression of the various packaging genes. The cassettes have been carefully designed to decrease homology and minimize the chances of recombination between them. This greatly reduces the possibility of generating replication-competent lentiviruses, thus making the system safer. This slide shows that our LV system outperforms those of our competitors, both in terms of high infectious titers and in terms of the lower proportion of empty capsids. This is true for both adherent and suspension HEC-293 producer cells. Our plasmids are now available at research, clinical and commercial GMP grade, and are manufactured using a regulatory-friendly, antibiotic-free process, all the way from initial clone selection through to bacterial banking and GMP manufacture. In the second part of my talk, I'm going to tell you about WuXi ATU's Plasmid production platform. Together with our colleagues at WuXi ATU, we offer a range of services to support our customers with every stage of their journey from discovery to product commercialization. These services include plasmid design and optimization, process development, cGMP plasmid manufacturing, and additional support with quality control, assurance, and registration. We have state-of-the-art analytical laboratories and can provide a comprehensive list of analytical services to support clients' product development and commercialization activities, including raw material and in-process testing, analytical method development, cGMP compliant validation, and ICH compliant stability testing. We are investing in equipment and facilities to increase plasmid manufacturing scale in the first half of 2022. When they come online, we will be able to provide from 50 milligrams up to 50 grams per batch to meet clients' requirements from clinical to commercial cGMP manufacturing. We also offer cGMP compliant E. coli seed cell banking as a service, as shown on the left. This feeds into the plasmid production process shown on the right. Fermentation is followed by cell harvesting and lysis, and then by a 3-step plasmid purification, and finally formulation and aseptic filling. WuXi ATU's facilities have been designed and built based on US, European Union, and Chinese cGMP requirements, with separate personnel and material flows and strict environmental control to avoid cross-contamination. The facilities utilize single-use closed bioreactors and critical disposable consumables to serve projects on a campaign basis. WuXi cGMP analytical laboratories provide a comprehensive array of analytical services to support the cGMP manufacturing of plasmid products through the full process. WuXi ATU has established a robust and systematic quality management system that meets or exceeds global regulatory standards. Each manufacturing facility shares the same quality system, and all have been designed to meet US, EU, NMPA and other major HA standards. We have put quality systems in place to maintain compliance and also to continue supporting our clients in an evolving regulatory landscape and throughout the product development lifecycle. There are many advantages to using WuXi ATU's plasmid production platform. It is versatile. We offer a range of batch sizes from 50 milligrams to 50 grams. It is quick, with turnaround times of 2 to 4 months. It is economical, with a high turnover rate and compact manufacturing footprint. The product quality is high, from an antibiotic and animal component-free process. And finally, we offer regulatory support with systems designed to meet global standards. We are proud of our viral vectors and plasmid production processes. They are the culmination of hard work by various different teams, especially Ping Zhou and the plasmid manufacturing team at WuXi ATU, and at OXGENE, the plasmid engineering and production, the viral vector design and discovery, and the antibody and viral vector manufacturing teams. And special thanks to Alyssa Bray, who is instrumental in the AAV vector development, Katie Roberts and Sophie Lutter for helping to prepare this webinar, And many thanks to Kat, Pella, Chen, and Ryan. And thank you for listening.
Okay, great. So the first question is, how long is a custom cloning project for a research-grade plasmid likely to take?
Thanks for the question. That depends on a lot of variables, such as whether the project will involve DNA synthesis or simply subcloning from an existing vector. It depends on how long the custom components are, such as the promoter, the coding sequence, and any other regulatory elements, and whether the custom elements contain any regions of very high GC content, for example, or secondary structure. So it could take as little as 2 weeks or perhaps as long as 6 to 8 weeks.
And if I work with OXGENE at the discovery phase, does cloning need to be repeated during GMP manufacture at WuXi ATU?
No. A construct that is assembled at OXGENE could be easily transferred to WuXi ATU for retransformation and GMP plasmid production.
Okay, so thanks. And just as a reminder, you can enter your questions at any time. We are running out of time for this webcast, though, so we just have time for one more question. If we don't get to your question, we're going to be passing those along to Richard. So please go ahead and continue to enter your questions in the question and answer window. So the last question for this webcast is, does RepCap optimization improve performance for all serotypes tested, and would it also benefit custom plasmids?
So all of the serotypes that we've tested so far have been improved by using our modified RepCap configuration. And so I would predict that it would also benefit custom capsid genes as well.
Okay, well, thank you, Richard.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.